Okay, welcome back. We are in block 3 of cell biology. I wanted to give a landscape view of cell biology and then aspects of molecular biology, which I teach as well. So in blue, this would be, especially here, this would, ooh, wonder why I did that. Uh, okay, let me do this. No. Hey, I want this. Okay. Well, I don't know why it's not working. Isn't that interesting? Huh. I don't know. Huh. Well, anyway, we have the protein. We've talked about protein interactions. We've talked about two hybrid, GST, protein studies, and manipulations. Aspects of cell and molecular are here, various mutations. We have certainly talked about tagging and immunofluorescence here. And then on the gene side, this is a lot of material we will cover in molecular biology. So I just wanted you to see the big picture here on sequence homology insights into structure and function in diseases. So it's kind of the gene side, the protein side of any kind of process. Block 3, we're going to hit membrane structure first. Some of this is review, some of this is probably brand new. Probably since high school, you've known fluid mosaic model that the lipids are fluid and it's a bilayer. Here's two layers of lipids. There's hydrophobic inside here and hydrophilic head groups pointing outward here, and there are proteins that are transmembrane proteins. It's about 20 amino acids that are required to stretch a typical lipid bilayer here. So this little clear area right here where I'm pointing to is about 20 amino acids. There are proteins that are peripheral that attach to other proteins here. And like I said, transmembrane would extend all the way through the lipid. And how do they know this is a structure? Well, they've actually done transmission electron microscopy and actually seen these structures here, and they know the size of them. They can see the lipids, the protein molecules here, and the association of proteins and lipids together. A simple lipid, usually in biochem, you start with a simple lipid, and this is a triglyceride. You may have heard triglycerides here. It's a 3-carbon backbone, and so this is 1, 2, 3 chains here. There's 16 CH2s, 14 and 14 here, and this is a long carbon hydrogen strings here. I guess you could call them strings. There are in lipids, phospholipids. So one of these carbons has been replaced by a phosphate, so it makes it more, has energy. And a lot of times on the phosphate, there is some type of other additive, maybe even an amino acid here. At this point, this is showing a little bit of the complexity of phospholipids. So you have lipids based on a triglyceride backbone, and then you also have a steroid such as cholesterol, which has a different structure. And cholesterol behaves differently than phospholipids and triglycerides here. We'll talk a little bit about that. And it just shows the proteins associating with the lipids. Certainly, proteins have to have at least 20 hydrophobic amino acids to span a lipid bilayer. There are isoprene chains. These are at these points are the carbons, and then the hydrogens are sticking out. And then if there is a COOH motif at the end, that's said to be a fatty acid. This makes it acidic here. So you have steroids such as cholesterol. That's its structure. We've talked about phospholipids, and here is a serine, phosphatidylserine. There's actually a serine amino acid here. Okay, so there's other amino acids and that can be added as well. And then there are fats or triglycerides here. Here's the 3-carbon backbone, and then the isoprene chains here. So 3 different flavors of lipid molecules. Most lipids and proteins, they will be adrift, and there's very fast lateral movement and spinning that's occurring. And then every once in a while, a lipid on one leaf flips to the second leaf, and that's called a flip-flop. In these isoprene fatty acid chains, if there are double bonds, those are said to be unsaturated. That means they're not completely filled with hydrogen. And notice the spaces there, it's more fluid. Versus chains that have no double bonds, they're said to be saturated here. And then buried in the lipid bilayer are cholesterol molecules. And cholesterol actually will lower fluidity, and there actually helps us our cells freezing at low temperatures, preventing the tight packing of phospholipids in a low temperature situation. So cholesterol is actually good for you. Okay. So phospholipids are the main structural elements in a lipid bilayer. They're shown here, the hydrophilic, water-loving head group, and the hydrophobic tails, fatty acid tails. So a molecule that has multiple characteristics called amphipathic. It can be either based on charges or based on hydrophilic, hydrophobic. Lipids will naturally, if placed in water, arrange themselves in a bilayer. And it's held together by those hydrophobic interactions. So these make them stick together. Okay, and this is just a space filling model. Here's phospholipids, and probably in chemistry you have space filling models. Here's the formula. This may be the schematic, and this might be the icon. So it's different ways to see these. But here's the polar head group. It's hydrophilic, water-loving. Here's the phosphoserine sitting on the what used to be the triglyceride. Now it's just 2 carbons, 2 fatty acid chains. See there's the COOH moiety, well, what's left of it there, and then the 3rd carbon has the phosphate and the serine attached. So it's a triglyceride that has the serine and the phosphate attached here. And this just kind of shows, here's the nonpolar hydrophobic cholesterol, and these are said to be steroid rings with a tail, and they're just showing the structure there. There's phosphatidylcholine. We've seen phosphatidylserine, and there's some other ones, ethanolamine, and I forgot the other ones, but there's multiple flavors of phospholipids here, which is interesting because the proportion of the different flavors of phospholipids is different in organelles and in the cell. So it's kind of one of these big secrets here. What is going on here in the structure? of organelle membranes. Here's the double bond right there. Phosphatidylcholine, and the lipid composition of any plasma membrane affects its fluidity. If it has a lot of fatty acid tails that have a double bond, it's going to be more fluid. And membranes containing a high amount of phospholipids with unsaturated chains are more fluid and less ordered, and produce the kinks, and you don't get a packing together, right? And so they're more spread apart. Lipids containing this type of phospholipid is thinner than most unsaturated. So there can be some different structural characteristics depending on the phospholipids. And here's cholesterol. It's shown here that's very much related to steroids. Here's a rigid steroid ring structure with a nonpolar hydrophobic hydrocarbon tail here. So it does have up here a small polar head group. And it can interact with other phospholipids. So cholesterol can, through the polar head group, interact with other phospholipids. And cholesterol actually prevents, like I said, the freezing of the membranes. It can be embedded in different areas of our membranes and resists packing. Here's just an interesting graph showing on the x-axis temperature going from 0 degrees freezing all the way up to 30. Ours is at 37. And then there's a permeability of glycerol. So you're measuring the amount of glycerol outside versus inside different membranes. And then they compare membranes with no cholesterol, membranes with 20% cholesterol, membranes with 50%. And you see permeability is very high when you have no cholesterol. Okay, so a lot more material could pass through our lipid bilayer here. Notice the amount of permeability goes down with the higher the cholesterol. So adding cholesterol decreases permeability. And there's a temperature aspect as well. So the higher the temperature, the more fluid, even with 50%, but it's nothing like having any no cholesterol there. And like I said, putting phospholipids in the water, they will tend to form this mycel structure. They'll actually order themselves in a circular way, and their space and material can be caught on the inside here. There's the lipid bilayer there. Talked about that. Lipids do not exist in a plane, energetically unfavorable. So this is some biophysics here, bioenergetics. So the sealed lipid bilayer compartment is the most efficient and energy favorable. So for making an artificial cell, we will certainly, this will work to our advantage. We can open up the lipids, add chromosomes, materials in, and then close them, and we can make an artificial cell. How do they know these lipids and what they look like? Well, you can visualize with a transmission electron microscope here. Okay. And there's this little easy device where you can actually have a chamber and you have lipids stretched across an opening, and you can add material on one side and test for that material going across. So some of the permeability experiments, you just change the nature of the membrane, and you could do all sorts of testing here. moving from one side to the other. Simple experiment here. And we talked about diffusion and versus in rotation versus the actual one lipid flipping to the other leaf. That's rare. Showing the unsaturated are less packed than saturated. Okay. And here's our friend the cholesterol, and showing the head group. This is an area in the membrane that would be, if it had a cholesterol, would be a stiffened region, and then there's the most fluid region would be right in this area here. And it puts itself, cholesterol embeds in this orientation because it has a polar head group and a hydrophobic tail. So here are all the different phospholipids. Phosphatidylethanolamine, not you have to memorize the structures here, probably in biochemistry you do, I know I did. Phosphatidylserine, choline, and of course to trick you, instead of calling it a phosphatidyl and giving this a name, we actually call this structure a sphingomyelin, even though it's the same thing in with phosphorus, but it just has this structure here, and it's a sphingomyelin structure here, and it has, instead of a oxygen, it has an NH group right here, and so that makes it a sphingomyelin, but that's really, sphingomyelin is just another type of phospholipid, but it has a little bit different structure there. So if you look in our body and look at lipid by weight, the amount of cholesterol varies, the amount of all the different phospholipids, ethanolamine, serine, choline, and sphingomyelin changes. From the liver to the red blood cells to our muscle fibers to the mitochondria to the ER, and then here's bacteria. Okay, so bacteria has 70% ethanolamine and just a trace of serine. So notice that same kind of trend. Not a lot of phosphatidylserine, but more than in bacteria. So here this is. Why is that? What's going on there? And notice bacteria don't tend to have phosphatidylcholine, but a lot of our cells have a nice chunk of it. Why? So there's some questions here. But anyway, just remember that the composition of the lipids can be different between tissues and probably even organelles. Okay. How do material get through this lipid bilayer? Sometimes there are pores, so it shows number 1, pores. And probably in your inquiry you talked about the nuclear pore. Proteins, DNA, RNA can go through. There is a size limit. So you have proteins that can be made in the cytoplasm on ribosomes, and some of these proteins have a zip code to go through the nuclear pore. Some have chloroplasts, some have mitochondria, some have peroxisome signals, some have signals to go into the ER, and once they're there, they're in the secretory pathway, and vesicles carry material. So you can go through pores, you can transport across membranes, simple diffusion, small molecules, amino acids and sugars. Large material probably has to be carried with a pore or channel. And then here's vesicular transport that we will talk about later. Here's the different organelles, and you've seen this since high school. Okay. Here's the nucleus. Ooh, it's moving around. It's very prominent in the cell. That's why when we break cells open, a lot of times we'll take the nuclear pellet and separate it from the rest of the material in the cell. The nuclear membrane is the content is located in the nuclear lumen and is surrounded by a nuclear envelope, which is an inner and outer membrane. And the outer membrane does have connections to the ER, and there are nuclear pores that allow material in and out. The inner membrane is called the nuclear lamin, which binds to chromosome material, chromatin, and other components. So there's kind of the structure there. And these pores themselves are made up of very specialized protein. It's about 120 million Daltons, so it's a big protein machine here that allows material in and out. There are 30 different proteins found in a pore. Basic subunit repeated 16 times. And there's a movement through these pores. There's no motor proteins. There are a ring of proteins that are the basket that they're still trying to figure out what that cage is. How do we know this structure? The transmission electron microscope actually shows this kind of pore structure here. And so transport factors with cargo come through, and if you're going across a gradient to a gradient, makes it energetically favorable if there's a lot here and a little there. But there are material that we want to go into the nucleus, and it has to go through this structure here, and we'll talk more about that. And inside the nucleus there's a nucleolus, also known as a nuclear organizing region, which is a stretch of DNA and ribosomal genes, the NOR region. Ribosomal proteins are made in a cytoplasm. They have a nuclear localization signal, and they come in and they interact with the ribosomal RNA, forming immature ribosomes that then leave and assemble in the cytoplasm. You're saying, well, why don't just ribosomal RNA go out and find? I think having a concentrated area allows efficiency of this assembly. If you put ribosomal RNA out in a cytoplasm, it's harder to get together with the ribosomal proteins. So it's a particle. Particle is nucleic acid with proteins. Here's the ER, endoplasmic reticulum. You have these, there's a nuclear envelope, and right there is the ER with the ribosomes. So there are two flavors of ribosomes that I'm sure you know. Cytoplasmic ribosomes, and they're ribosomes attached to the ER. They call that the rough endoplasmic reticulum, because that's how it looked to people that were viewing the ER. That is where proteins that are destined to be secreted or moved into the different biosynthetic pathway, they have to have the information to get into the endoplasmic reticulum. So the ribosomes that are attached have to open up channels and allow these proteins to go inside the endoplasmic reticulum there. So there's a whole process that's required to move into the ER. ER has enzymes that hang around, and they help proteins fold. Drug detoxification. The smooth ER with no ribosomes, a lot of lipids are made there, so it's an area of lipid biosynthesis. Cholesterol and fatty acid metabolism is also made there. So these are the workspaces of the cell for a lot of materials. It's also the beginning of biosynthetic pathway and protein sorting. And there's an actual sequence of amino acids called the signal sequence. And it has certain characteristics that we'll talk about. The membrane proteins are inserted in the right orientation, and secreted proteins move into the ER and then move through the Golgi and then maybe to wherever they're going. So rough and smooth, we talked about that. The smooth ER is enzymes that help detoxify. It's a workhorse in the liver. And it talks about other properties in other tissues and whatnot. So the ER manufactures membranes and secretory proteins. In the pancreas, the ER produces the insulin, which is good. White blood cells, it helps make antibodies. And how does this work? Well, this little cartoon, and we'll go into more detail later. Here's the messenger RNA coming in here, and the ribosomes are associating, and this little darker green is the protein actually going through the ribosomal pore there, and then the protein can be released and go to mitochondria, chloroplast, peroxisomes, nucleus. However, there is another pool of ribosomes that attach to the ER surface, and notice the structure of the protein. There's a red structure, and that is called the signal sequence. So if a protein in its N-terminus has the signal sequence, that is detected by the ribosome, and that signal sequence actually allows it to attach the membrane, and it's shown here, and that goes through the pore or channels, and that brings the protein inside. And the signal sequence can be clipped, or if this protein is a membrane attached, the signal sequence can serve as an anchor. Lots of different things that can happen in this process here, but the protein has to have a signal sequence. So experimentally then, you should know this, how would you prove that a protein signal sequence, ER signal sequence, was required to get into the ER? Yes, you would mutate it, get rid of it, and that then say do a Western blot. You know, here's the ER fraction, here's everything else. The protein is everywhere else, not in the ER. If that's a signal sequence, it would be a lot of it would be in the ER. They've also taken green fluorescent or luciferase and had the gene for those, and they actually added the extra genetic material for a signal sequence, and lo and behold, the GFP ends up inside the ER. So this signal sequence is necessary and sufficient for movement into the ER here. Here's our friend the mitochondria. Looks like little red hots, little tubes there. This is just fluorescence staining. And it may be more this kind of superstructure. This is in muscle fibers, or this is sperm tail here that have muscle fibers. Allows the sperm to move for fertilization. And of course, lots of mitochondria in high energetic tissues and less and lesser energetic tissues. One of the richest sources for mitochondria is in probably hummingbird flight muscle. If you didn't know, hummingbirds move their muscles very quickly and they need energy. And probably in your inquiry you found that the mitochondria is a double membrane organelle, and the inner membrane is folded to form cisternae, or cisternae, I guess. And this is the structure it looks like. Glycolysis, Krebs cycle, electron transport, a lot of that material is occurring in the organelle here. So having a protein targeted to the mitochondria, again, where does it go? Does it hit the membrane and stop? Does it go into the outer space? Does it go into the inner membrane? Does it go into the matrix, the space inside the folded layer here? Here's the signal sequence, probably in your inquiry. Here's the precursor protein. So when you see the word precursor, that means something's going to change that precursor to the mature form. Okay, so that is just two words for this material you need to understand. We'll talk about that later. So precursor is a protein that it needs to be changed to become a mature form. So here's the precursor. They found the N-terminal signal sequence, and you're saying, didn't we see that for the endoplasmic reticulum? Answer is yes, we did, but it's different. And there's actually a receptor that detects that, and that receptor complex binds to other proteins, protein translocators, and you end up getting a contact site between the inner membrane and the outer membrane, and this mitochondrial protein goes inside. It looks like a pretzel, and in this case the signal sequence is cleaved at that point, and the mature protein is made, little pretzels made inside the ER. Again, you can stop, embed into the membrane. You can be moved into this space, moved into this membrane, or moved here. So I think we'll stop here, and we'll continue a little bit more in depth with the Golgi complex and lipids and lipid structure here.